Cardiac looping is a crucial process in the early development of the heart, transforming the linear heart tube into a more complex structure that begins to resemble the mature heart. This video will explore how cardiac looping positions the future heart chambers in their correct anatomical locations, setting the stage for the subsequent septation and formation of a fully functional heart. Before cardiac looping, the heart starts as a simple, straight tube, differentiated into regions like the sinus venosus, primitive atrium, primitive ventricle, bulbus cordis, and truncus arteriosus. These regions will eventually develop into the different chambers and outflow tracts of the heart. Cardiac looping begins with the bending of the heart tube to the right, a process known as dextrolooping delooping. This rightward curvature is critical for establishing the correct spatial relationships between the future heart chambers. The direction of looping is guided by the asymmetry of the developing embryo, influenced by signaling pathways like the nodal and lefty genes, which establish left-right asymmetry. As the heart tube bends, the bulbus cordis, which will contribute to the right ventricle and outflow tracts, moves anteriorly and to the right, aligning the ventricles and outflow tracts properly. The primitive ventricle, which will become the left ventricle, shifts to the left and downward, ensuring correct ventricular positioning. The primitive atrium moves dorsally and cranially, positioning itself above the ventricles, which helps create the correct orientation for the future atria relative to the ventricles. The movement of the atrium above the ventricle leads to the formation of the atrioventricular (AV) junction, the future site of the mitral and tricuspid valves. Looping also aligns the truncus arteriosus, which will split into the aorta and pulmonary trunk and the conus cordis, setting the stage for the division of the outflow tract into separate pathways for systemic and pulmonary circulation. By the end of cardiac looping, the originally straight heart tube transforms into an S-shaped structure. This S-loop creates the basic layout of the heart, with the ventricles positioned side by side, the atria positioned superiorly, and the outflow tracts aligned for proper circulation. After looping, the heart is primed for septation, where the chambers are divided, and the outflow tracts are aligned for systemic and pulmonary circulation. Cardiac looping is crucial for ensuring that the heart's chambers and major blood vessels are positioned correctly relative to each other. Errors in this process can lead to congenital heart defects such as dextrocardia or transposition of the great arteries. The looping process sets the stage for septation, where the heart's chambers are divided by septa and the atrioventricular and outflow tracts are properly aligned. Cardiac looping is tightly regulated by genetic factors and signaling pathways, including the nodal, lefty, PIX2, and hand1 genes, which establish left-right asymmetry and guide the direction of looping. The cilia on embryonic cells play a role in establishing left-right asymmetry by generating fluid flow that influences gene expression. Cardiac looping is a vital process that transforms the heart tube into a looped structure, positioning the future heart chambers and outflow tracts correctly. This process is essential for the proper formation of a functional four-chambered heart. Understanding the intricacies of cardiac looping is crucial for recognizing how congenital heart defects arise and for developing strategies to diagnose and treat these conditions. Thank you for watching.